Hi, everyone. So this is not Wiley. This is your Vice President, Josh Black. I'm filling in for Wiley today. So welcome to the Sacramento Aquarium Society November meeting. I've got a bunch of announcements today. Hi, everyone. So this is not Wiley. This is your Vice. Got some echo there. First of all, I want to welcome the new and renewing members to the broadcast today. And also, if this is your first time attending, please feel free to join if you're really interested in what we do here and what you see today. Our website, I'll put up a link to our website, and you just want to click on the membership icon, and then you'll be free to join. So next, I'll announce our raffle winners for this month, and I apologize in advance for not pronouncing the names correctly, but I'll try my best. Your winners this month are going to be Michael Zigachenko and Lee Friedman. And again, that's Michael Zigachenko and Lee Friedman. And if please contact, or I guess Modesta will contact you very soon about that. Also, some great news. Our former secretary and board member, Dan Curran, is doing much better. We appreciate all your thoughts. And please continue to, keep Stan Moray in your thoughts as he battles through his health issues. Next, we had an election today, and I'm going to tell you all the results of that election. So I'll do a roll call. And so our president is still Wiley Peck. Vice president is still myself. Treasurer is now Jonathan Wardrip. Our secretary is Donna Laird. And then our board of governors consists of Aaron Markley, Willie Markley, Matt Adams, Ryan Enright, Michael Pohl, Jan Nolta, and Larry Schenkel. So thank you to all that participated in the voting and nominations. And that is your new board of governors and your president, vice president, and committees. Okay, so next, I'm going to talk about our sponsors. So I'd like to thank our sponsors. So our local sponsors are Splash Aquarium, Aqualife Aquarium, and Exotic Aquarium. And just a reminder that Aqualife Aquarium gives you 10% off of all purchases if you show them your membership card. So that's a pretty nice benefit for us. And I also want to mention, yes, Lee's Feed is also a local sponsor. And please support our sponsors as they support us financially. We really appreciate their support. Actually, sorry, they don't support us financially, as they support us, and please support them. Apologize for that. Again, I want to mention our Teespring store. So right now we have a 20% off promo code, and that's promo code is now up on the screen. And so it is good until midnight of tomorrow or Sunday. And yeah, the Teespring store now has a bunch of different logos and decals, which is really cool. We've got t-shirts, hoodies, mugs, neck gaiters, masks, even dog hoodies, believe it or not. So all kinds of cool stuff there. So feel free, if you're interested, check out our Teespring store. And I'll be putting in the links for the Teespring store inside the chat over time here. And we appreciate all the support. And if you feel like donating, you're welcome to donate at our, there's our website, sacramentoaquariumsociety.info/donations. And let me see if there's anything else. I believe that's it. So again, welcome. And next, we're really excited for this speaker, Brendan Ebner, coming all the way from Australia, speaking to us about clean gobies. And I'm very excited because this is one of the first times I can remember that we're getting a speaker to talk about gobies. And I guess I'll throw it over to Michael to introduce Eb, as well as Eric Thomas.
And so what's up, everyone? Like I said, just wanted to pop in here for just a second. And that side of me is Eb, and then I'm Michael. And we're going to go ahead and throw Eb's presentation up. He pre-recorded a presentation, and then he's going to be around for about a half hour after the presentation for Q&A. So if you have questions, be sure to write them down so that we do some good Q&A. And so we're going to go ahead and go down and bring the presentation up.
Hi everybody, my name's Eb. I'm from the department called TropWATER at James Cook University here in Far North Queensland. I also work as a visiting scientist to CSIRO Land and Water, which is our sort of federal science organisation here in Australia. Anyway, a big thank you to the Sacramento Aquarium Society for having me along this month. I'm really looking forward to telling you a little bit about some of our wetter streams in the tropical part of Australia. In particular, I'm going to talk to you about these short, steep coastal streams, and they're in sort of the wettest of the wet places that we have, and probably something that might be comparable would be some of the systems in Hawaii or maybe in, in the Caribbean, those sort of places in Indonesia. So they're really kind of wet, high rainfall, tropical rainforest type areas. In fact, I could probably give you a bit of a look at what some of these streams look like. So this is Noah Creek on a rainy day, and this is a really low gradient part of the stream. So even though this isn't the steepest part of these sort of systems, you can see there's still a fair bit of water moving down through these streams. And this is the sort of stuff we're snorkelling in the riffles and the runs, and also in the pools, and then climbing up above the waterfalls and what have you. So I'll step you through some of this stuff today, and I'll probably talk really about sort of your typical looking fish that are sort of, pan-shaped, swimming scaled fishes, the larger bodied ones, in particular the jungle perch and the silver grunters that we have in these streams. And then I'd like to introduce you to some of the smaller bodied cling gobies, which are a real feature of the short, steep coastal streams. And then finish off with the sort of freshwater moray, which I guess is the flagship species of the region, and something that more and more is finding its way into the aquarium trade. And I imagine a few of you have either kept them already or you're interested in the idea of doing so. Okay, just a little bit of basic background. So I'm sure all of us are familiar with elevational gradients, and if you go to the seashore, you see within just a few centimetres or a few feet of the tidal limits, you see this zonation that occurs of different fauna and plants and how they sort of comprise the seashore. And then at much bigger scales along mountain ranges, such as in the Andes, you see that we're all sort of familiar with the idea that there are animals and plants that live at different elevations because of some of the biophysical properties of those systems. Now, in streams, there's been a fair bit of work done, particularly in Northern Australia and in Europe along some of these gradients. About maybe 15 years ago, I was involved in some work in the colder parts of Australia, in the Murrumbidgee River, and we did some sampling of fish by electrofishing along 13 kilometres of the Murrumbidgee River. And it sort of produces this sort of data, which turns probably a lot of you off watching this stuff. But if you just look at the bottom graph, you'll see a series of sort of barcodes, which is showing you 160 different electrofishing shots consecutively that we took over a few weeks. And the little pixels that you see there for the distribution of the 7 types of fish with their Latin names on the y-axis is just showing you that some species are a lot more common than others. So some of them are turning up in pretty much every electrofishing shot, one after the other with a few gaps. And then you notice that there's a few species that are much rarer than that, and they're only distributed along the river in patchy fashion. Anyway, we did this work a fair while ago, which basically copies what the Americans had done for years and years, and it was nice to have an Australian example. And I was really licking my lips at the idea of moving to the Australian tropics and repeating this sort of process. And when we did move to the tropics eventually, my family and what have you, my son came out with me and did a bit of snorkelling work. And in some of these smaller streams where there are no crocodiles, we're actually able to snorkel the entire catchment. So starting down at the tidal limit and then just slowly working our way up into the hills. And that gives us the same sort of data that we'd had from those sort of electrofishing studies, in the sense of giving you an idea of every 100 metres what the fish assemblage looks like, and as it sort of progresses up through the mountains. Anyway, I'm going to talk to you a little bit about some of that work. So these streams are in a part, as I said, called the Wet Tropics, and some of you who have been to Australia have probably been to the Barrier Reef. You can see in the middle of that graph there on your left-hand side, the city of Cairns, which is a major international tourist destination, or it was so prior to COVID. And the streams that I'm talking about are within about 50 or 60 miles of Cairns. So Noah Creek up there to the north, a little creek called Turtle Creek, which is sort of just north of Cairns, and then a third one called Paul's Pocket Creek, which is sort of on some pretty steep country going straight into the ocean. Anyway, we snorkeled these streams from source, or from down at the tidal limit up to their source in the hills. Here's just a graph showing you that the steep elevation of these streams. So down on the x-axis here, we come up to the number of 3,000. So that's 3,000 metres in terms of how long the stream is, which is 3 kilometres, or a couple of miles in your terms. And then on the elevations, you see we're getting up to a few hundred metres elevation within just 1 or 2 miles of the ocean. So really steep systems, as I said, like Hawaii. And if you look at the picture here, young James is actually in that picture, a bit of a Where's Wally. He's sort of scaling that 5 metre high waterfall there. And it gives you an idea of what we're talking about when I'm mentioning these large waterfalls or barriers in the system. So just returning to the graph there, you'll see that each of these streams has barriers in different places in terms of their distance from the stream. And as you're about to see, that has a pretty major impact on the large-bodied fish, which have to migrate up through these systems, a little bit like your salmon runs over in the US. Here's just a couple of graphs from some of this work. Just on the bottom axis are scientific names of different Gobies species, and you can see that these species tend to specialise in specific elevations along the stream. So there's some that are down in the lowland areas near the tidal mark, and then as you step up through the system, you get these specialists that are only found above waterfalls at high elevation. And then the one there that's called Sikiopteris lagocephalus is the rabbit-headed goby. It's you can see it's got a bar that basically shows it's an elevational generalist, so it can be found down at the tidal limit, and it can be hundreds of metres high up in the catchments as well. So it's quite the all-rounder. Anyway, here's some more graphs, and you don't need to sort of be blown away by these, but basically on the y-axis, they've got counts of how many fish or prawns that we got in a 100-metre section, and going along the x-axis on the bottom on the horizontal is basically moving from 100 metres from the tidal limit, 200 metres, 300 metres, etc., up to maybe 3,000 metres or so from the tidal mark. And each of those bars indicates in black the counts of these jungle perch, which are the big-bodied fish which are predators. And you can see them in the picture there in the top right. They're the jungle perch. And you see that where those bars are, there aren't any grey bars, which is indicating the prawn counts and the shrimp counts that we got. And you'll notice that once those black bars drop out, which is the counts of those jungle perch, then we get really large counts of prawns and shrimps showing up. And, that's no coincidence really, that when the predators aren't there, that the prey are actually really conspicuous out in the open and quite abundant. And the third picture down the bottom there below the prawn is a little cling goby that we call the mustache goby. And this study was the first time that species had ever been recorded in Australia. It's also found in sort of the Solomon Islands, Papua, and some of the more equatorial high islands in the Central Pacific. But these studies kind of led to the first record of this mustache goby above waterfalls, which is kind of cool. Okay, so now what we're doing here is just having a quick chat about that. So these sort of findings are similar to results that have been found in the Caribbean and in the Central Pacific, but this was the first records of sort of continental Australia where this phenomenon was happening, and it's kind of neat. So now what I'd like to do is just talk a little bit more about the cling gobies rather than the large-bodied fish, i.e. the jungle perch and the prawns. And so here we have the mustache goby, which is on your left, and on the right we have another very closely related species, which is, its scientific name is Smilosciopus felmani, and so both of them are co-occurring above a waterfall in the absence of any of the big predators. Okay, so after we'd done these 3 streams, and some of that work was done sort of over probably about a decade ago, in conjunction with some of the Indigenous ranger groups in the region, we then got a grant through the state government in Queensland, through Everyone's Environments grant, and that allowed us to go and survey another 10 or 12 of these streams in a rapid fashion to get a better handle on where these cling gobies were in Australia. So just by way of a little bit of background, there's sort of 3 main running waterway types in the Australian wet tropics. There's the really big, large floodplain rivers. These have often got crocodiles in them, and really big crocodiles, so they're not a system that you'd be snorkelling in. You'd be using boat electrofishing or netting in those sort of systems to sample fish. Then we have some small coastal streams which are more swampy and really flat, not fast-flowing streams except during the wet season. And there'd been a fair bit of survey work done in these first 2 types of waterway. When I moved here 12 years ago, there really hadn't been much work at all done in this 3rd type of waterway, which is the short, steep coastal streams. So that's specifically what this grant was for, a year of survey work in those systems. Just by way of some general background, there's a whole bunch of different government and non-government managers that operate in these sorts of catchments, ranging from Indigenous people to the National Parks Office, surf lifesaving clubs, farmers, schools, people responsible for waterways, threatened species, the Great Barrier Reef, Marine Park Authority, etc., etc. So multiple managers and jurisdictions are interested in these sort of systems, and a whole bunch of different user groups in terms of people from the public that actually use these catchments. A lot of them are absolutely stunning, pretty systems, which are really steep, and so they're places that people go for wedding photos, stay in resorts, have a swim on the beach, go for, lovely walks, do a bit of swimming at waterfalls, this sort of thing. But they're, in some cases, they're also privately owned, where there's farmers operating in the area, and in some cases people are fishing and what have you. So a real mixture of people use these habitats, whereas some of those other systems that I'd mentioned earlier in terms of waterways are in much flatter catchments, which are open to being used by agriculture and urban development and the like by people. Whereas these short, steep coastal catchments, because of the nature of their steepness, are often not sort of occupied by people in terms of living dwellings. Here's just some pictures of some of the creek mouths of the systems that we're talking about. And so if you look at them closely, you'll realise they don't actually have mangroves associated with them. They don't generally have estuaries with any kind of brackish water, and that's because they're high rainfall areas and the water comes down off the mountains and carves out through the sandy beaches and goes straight to the ocean. And this creates opportunity for quite different aquatic fauna living in these systems because there isn't that influence of the mangroves and the brackish water. The 2 maps that I've got up there are just of the wet tropics, and that wet tropics region on the right-hand side, you'll see a whole bunch of green shading, which is the World Heritage Area and National Park. And so that's where a lot of freshwater fish work had been done over the years. And then as we came, if you move over onto this map on the far left-hand side, you see a little bit of yellow shading just at the coastal edges. So they're the systems that I'm talking about. They're only 3, 4, 5 kilometres long, a lot of these creeks, really steep areas, and they're not sort of spread throughout the entire wet tropics area. During this project that we had for a year, we came across a number of pest fish and pest species, but particularly cane toads and their tadpole phase, obviously a species that's come across from South America. And then we had pigs and in some cases tilapia in a lot of the neighbouring catchments, but we didn't actually find them in the short, steep coastal streams, which is good. You might notice here from the table that a number, probably half of the streams that we surveyed had some form of water extraction from humans going on, whether they be small dams and weirs or roadway crews pumping water from them late in the dry season for either fire management or building road surfaces, that sort of thing. So there's really a human element in terms of how these systems are being engaged by people as well. Okay, so it's a lot of background. I'm sorry to put you through all of it, but it gives you a bit of context. Here we're into some of the beautiful cling gobies that I'm actually talking about. These photographs are of the males of each of these species. So up in the top left, starting with A, we've got birdsong's cling goby. That little species is only about 3 centimetres long. So it's a really tiny species. The male is bright red and very beautiful with elegant little scales. The next one, the orange cling goby, you can see here with the blue face, absolutely stunning male species, often found very close to the high tide mark, just above the tidal limits is sort of where it often is. It's the first one you encounter on your way up the hill. And then the one with the blue face, top right, we call the emerald cling goby. It often has an emerald green colour throughout the year when it's not displaying. And then when it comes into the courtship period, it gets this really bright blue face and a black body. Sometimes it has white stripes and sometimes it's just jet black. The one in panel D that you'll see in the middle on the far left is the one we call the opal cling goby. This one's listed as critically endangered in Australia. However, it is found and is fairly widespread in the central Pacific. So it's in Solomon Islands, it's in Papua New Guinea and Indonesia, Sulawesi, these sorts of areas. Okay, and then we've got a few of these E and F, the Smilosicyopus genus, and these individuals are found up above your first waterfall. They're often in very small numbers, maybe just a male-female pair. They have tiny little mouths and they actually hunt macroinvertebrates. They're little micro-predators, whereas the first 4 species that I introduced are all herbivores that graze on biofilms on the rocks. We come to G there. That's a stunning species. Its science name is Sicyopus discordipinus. It has this beautiful red flag tail and body there that the males use to signal to each other, much like skinks do. And then H is the rabbit-headed cling goby. It's the one I mentioned earlier that is a real elevational generalist. It's found throughout these short, steep coastal streams. It's our most widespread species in the Australian wet tropics in terms of cling gobies. And then letter I, the very final one in the bottom right corner, is a really large cling goby. It gets to maybe 15 or 20 centimetres long, whereas most of these other species are only sort of 3, 5, 6 centimetres long. They're quite small species. So I is cyanus, we call it the ghost cling goby. I'm just trying to remember its name, sorry. It's Sicyopteris cynocephalus. It's a stunning animal, and we've only found it in 1 stream so far in the Australian wet tropics. So it just gives you a bit of a look at the animals we're talking about. And as I said before, they're found along these elevational gradients with that blue red-tailed one, the rabbit-headed cling goby, pretty much throughout the whole stream elevation, and then other species sort of sprinkled and stepping their way up through the catchment. Okay, here's some of the mathematical modelling that was done from the work that we did in a package called MaxAnt, and this work allowed us to survey different regions along the wet tropics coastline in some wet and some dry areas. which then sort of fed back into the model to give us an idea of the true or the likely distribution of these species in Australia. And this graph here is really just showing us that we have these lowland specialist species, if you look down the bottom on the x-axis, and we have altitudinal generalists and upland specialists. And prior to us doing this study in 2015, you'll see that the grey bars indicate what we knew about or how many streams where we'd found these species previously. And then by doing this extra 12 or 13 streams, you see the black bars showed that we had an increase in the number of streams that we now recognise these Gobies from, quite rare species. And in particular, you see that we probably had a doubling in the detections of these high elevation specialists. And that's because sort of up until then, we hadn't done the work as systematically. We'd mainly focused on snorkelling in the bottom sections of streams close to roads and boat access areas, whereas because we did this study in a more systematic way, it meant that we had to put a lot more time into those high elevation areas. And even though we didn't find many fish, that's where you detect these much rarer high elevation specialists. This is just the results of the modelling work, and you probably can just see on the edge of these coastlines little red and yellow areas on the eastern seaboard, which shows where these species are distributed, and they're not distributed inland in that area that's blue, where most of the fish work had been done in the wet tropics up until then. We probably don't need to focus in on the modelling outputs here. No one's probably that interested in it, but it basically just tells you that these species are found very close to the ocean in those steep catchments. Anyway, here's the model data combined for all of the Gobies species. And what was fascinating after this project in 2015 that came out of it, and you might see in the middle figure there, the map, it's pre-January 2015. There's a fair bit of red and yellow and what have you, particularly in the northern section, the upper section of this map. Then when you flip over to post-January 2015, you see that red shading's been diminished somewhat. And actually what it essentially tells us is that these animals are very rare. They're only found in a few of these very small streams. And prior to the study, our model estimated that they were they had probably 2.4% of the entire wet tropics as favourable habitat. And by the end of our study, the model had been re-updated to just 1.5% of the region. So it showed basically that, you know, they were quite rare in the system, even rarer than we'd previously thought. This is our coastline, and I apologise, it's flipped. You can see the northerly arrow focusing out to the right-hand side. And it's basically, so we're looking up into the catchments there, and you see all the creeks that we sampled listed with lines going to where they were on the map at the coastline. And then this middle panel with the bubbles on it is basically every one of those bubbles, if it's big, means that we sort of got 20 or 30 or 50 of that type of Goby. And then when the bubbles are really, really small, it means we just got sort of 1 or 2 of those Gobies in a particular stream. This figure steps us through 9 different species of Goby that came out of the systematic work, and sort of shows it according to the altitudinal, elevational, lowland specialists and the upland specialists and the generalists. And it shows you on the bottom graph the catchment sizes, which are very, very small catchments. You see the biggest of these catchments is only 30 kilometres squared, and the majority of them are just a few kilometres squared. So very small stream catchments with high rainfall that allow them to persist with permanent water, and that's what these fish are sort of focused in on. So what do we do with that information? We calculate what amount of area in Australia is used by each of these species, and the numbers come out as very small, and then that allows us to sort of see whether some of these species should be listed as threatened species or vulnerable species under our national legislation. So that sort of has conservation ramifications. And there's probably only one species there, the rabbit-headed goby that I spoke about, and its science name, it's at the top there, Sikiopteris lagocephalus. It's the generalist that's widespread, has a big footprint, and it's the one that I think aquarists should be able to keep in Australia without too much harm to the environment because they have big populations and they're widespread. But currently I will say that all of these species in Australia are under our state government legislation in Queensland, they're no-take species. So we're not allowed to collect them and keep them as private aquarists in Australia. But I think maybe in time the legislation might change for that species at the top, as I said. There's a whole bunch of journal publications that have come out of some of this work. Those of you who are interested in following up on it, you can send me an email or track some of these papers down on the internet or whatever if you really want to know some of the details. Here's just a picture of two of the species Stiphodon rudalarius, two males displaying to each other. So in that picture, those fish are sort of 3 or 4 centimetres long, full-grown males that are just having a contest there, getting prepared to court with the ladies. Stunning little fish. And there's a few of my colleagues coming out doing some of the snorkelling in one of the streams late in the dry season when there's not too much flow. Okay, so I hope you've enjoyed that. And the next part of this talk will move on to the morays. I'll just see if I can show you a couple of videos which should complete this little part of the presentation. So this is just Noah Creek, in the lower parts of Noah Creek, just to give you a bit of a feel for what this habitat looks like. So fairly fast flowing, gets much faster flowing when you get higher up in the catchment. That gives you a bit of a feel for what one of these streams looks like. And I'll just actually, you might just pull up for a moment there. Okay, I'd just like to step you through a little bit of video of some of the animals I've talked about, just so you can sort of visualise them. Starting off here with the jungle perch. So the ones with the 2 black spots on their tails are jungle perch, and then you'll see every now and again there's another one that looks pretty similar, and it has sort of a red edge to the back of the tail. They're the marginate jungle perch. They're quite 2 closely related species, but by far the majority of these fish here are big jungle perch. They're about sort of 20 to 30 centimetres long. And every now and again there's a mangrove jack in there and some striped grunters. These are in the sort of the deeper pools in the short, steep coastal streams. They're often in the plunge pools. And you can see the water here sort of down deep there's not really any flow to it at all. They're sort of just sitting down in the boundary layer late in the dry season. And these are the animals that are sort of keeping those prawns and shrimps in check, at least during the daytime. At night, sometimes you get prawns and shrimps coming out where these species are present. So there's sort of a pretty strong interaction going on. Anyway, I just thought you might like a look at some of those bigger animals. Quite graceful swimmers. And I'll just pull up another video for you. Okay, so here's some footage of the little cling gobies, the opal cling goby, and I'll just open that with media player because the other package seems to be playing up a bit. Yeah, so this here is the opal cling goby. This is the male. It's about 4 centimetres long. This is some footage I took a year or 2 ago in a little tiny plunge pool. I dived with this male and female opal cling goby for a couple of days. They got used to me and I got used to them, and I sort of kept my distance most of the time. So I've really got the camera on a magnifier here to be able to zoom in. The water's really, really shallow. So, I mean, this water would be not much more than a foot deep. So my gut is getting caught on the rocks, and I'm sort of reaching forward to try and film these animals without annoying them. And you'll see this boy cling goby here, the opal cling goby. He is dancing around with his blue iridescent stripe, and he's catching whatever light he can get that's coming through the rainforest canopy and a little bit of my dive torch, and he's angling his body to throw that iridescent blue light towards the female as he dances around the high points in the stream. You'll see her in a moment. If you look really closely down in on the stream bed, you'll see a tiny little brown fish on the bottom. He just gave her a kiss then, and she started following him up towards where they're going to spawn, up in this little cave. So the boys' little cave is up here underneath these little fine tree roots, and that's just in maybe 10 or 15 centimetres of water, so it's very shallow. And he's just constantly displaying to her and getting closer and closer to her in tightly knit circles, and she's just making her way into the little cave there where they'll lay the eggs, and he'll fertilise them, and then he'll guard the embryos for the next 24 or 48 hours. Probably have a few hundred, maybe 1,000 embryos in one batch, and then after a couple of days they'll wash out to the sea as little hatched larvae, and they'll live in the ocean for a few months before returning to the streams and then growing into adults. So there's a bit of lightning here going on in the background. So for this final part of the talk, I just want to focus in on the freshwater moray, which is an absolutely stunning animal. And you'll see, I'll just show you a little bit of video of one in the wild. This is an adult and just getting surrounded by a few larger fish swimming around, the jungle perch that you were just introduced to, and a few mangrove jack at times. And you see, this is how you see the animal a lot of the time. It's just, you'll just see the head popping out from underneath the boulder, and they're really quite shy and fairly quiet sort of animal, not particularly aggressive and really wary of divers. Gymnothorax polyuridant. It gets to probably about, well, the biggest ones I've seen are sort of about 1.20 meters, but commonly in the stream you sort of see them around anything from maybe 20 centimetres up to about 80 centimetres long. That's probably the more common size that I've encountered. Anyway, Okay, so we'll just step through a little bit to do with their habitat use and feeding ecology, and then a little bit of aquarium care. But to start off with habitat, when I first moved to the wet tropics, 2008, 2009, there hadn't been too many records of this species from the region. And so I went about collecting records from other scientists that I knew that had at least 100 hours of field experience in a freshwater system and in an estuary system or in a nearshore marine system to see if anyone had detected this species. And if you look at the graph on the top left-hand side, you see that most of the records for these species, and probably half of these records are my own records, are around very close to the tidal mark, just on the upstream side of the high tide mark, in the first few 100 metres into the freshwater systems. We have 3 records at the time which were sort of for more than 10 or 20 kilometres downstream in some of the bigger river systems, so in sort of more brackish water. And we had 1 record from up to 12 kilometres upstream of the high tide mark in one of the systems. If you move to the graph that's on the top right, just looking at that data in a slightly different way, basically 27 of those records were in pure fresh water. 13 of the records were in estuarine situation, like brackish water, and then there were no records from any of the nearshore marine reefs. That's despite a huge amount of diving that goes on the Great Barrier Reef in the wet tropics region. So from these data, it seemed that this species, as a juvenile or an adult at least, is not using marine waters. And if we actually look to the graph down in the bottom right-hand side, you'll notice that when these same data records are plotted, that the few records that are in brackish conditions are actually in very low salinity conditions. So, 3 and 4 parts per thousand. So whilst they are in estuaries and brackish waters, they're very much preferring the fresh water and the only just brackish type water. You'll see in that graph that there is 1 record of an individual in pure salt water. And if you look at the x-axis on the bottom of these graphs, that's telling you how big these individual morays actually are. And so that 1 record that is in a marine system is an animal that's 8 or 9 centimetres long, So I'd argue that it's probably still kind of coming back from the ocean as a larva, and it's as part of its life cycle, it sort of hasn't really settled properly yet. But you can see by far the other 30 or 40 records that we've got here are of animals that are in essentially pure fresh water. So this is kind of interesting because it was the first serious evidence of 200 species of moray in the family Muranidae, that there was indeed a species that had a juvenile and adult phase that was essentially fresh water rather than being marine. So it's pretty exciting times. And Tsukamoto from Japan also brought out a paper where he looked at the otolith microchemistry of 4 of these eels of Gymnothorax polyrodon from Fiji, and that otolith microchemistry supported what we're saying here, which is that this species has a larval marine phase and then it settles as a juvenile and moves into fresh waters to exist. So back in the time, it was pretty exciting information. And then over the last 10 years, sort of had more to do with the animal. Whilst we've been doing clean Gobies surveys and other things, surveys, you come up against this species from time to time. Here's a few more recent records under bridges, under concrete bridges and boulders associated with Cooper Creek system. And we sort of published this work recently to show that some of these individuals that are down near the tidal mark do actually encounter maybe 10 or 15 parts per thousand salt at times on really high king tides. So whilst they are occupying the freshwater reaches, they can tolerate salt to some degree as well. These graphs here are just stepping through different attributes of about 30 records we had of this rare animal, this eel. And you don't need to freak out about these graphs if you're not familiar with these sort of indices. But just as a general guide, if any of these bars in these graphs go above the dotted line, that means that the species has a strong preference for that particular attribute. So in the first graph in the top left-hand corner, it's about flow velocity for the records that we had of this species. And what you see there is that these animals are selecting for very low flow, if at all any flow. So they're basically in 0 metres per second flow. If you move to the second graph in the top right, what we're seeing here is about substratum preference, and the 30 records we had show a really strong preference for boulders. That's their preferred resting habitat, underneath boulders. But you'll see that they're also using bedrock and cobbles to some extent, but their strong preferences for boulders. And then the third graph, which is down in the bottom left, is showing you the depths that we found these eels at. So pretty much every one of these records is an eel sitting under a boulder in no flow, and they're often in sort of about 1 metre depth of water, and in some cases maybe 2 metres depth. They're not in the really shallow parts of the pools. So as a general rule, they're in the deep fellweg, or the deepest sort of part of the channel in these short, steep coastal streams. So that just gives you a bit of an idea of what habitats they live in. They do go out and forage in the riffles and runs and the shallower parts of the pools, but these records are basically when the animal's sitting in its lair, just sort of resting and not feeding. Okay, I'm sure a few of you would like to know a little bit about the aquarium care. I'm guessing some of you in the society have already kept this animal before, and I hope you've had good fortune with it. When we did move them into aquaria initially, the objective was not only to keep the animals and enjoy them, and enjoy learning about them, but to kind of get a feel for the fact that they can live in fresh water. So you'll see in panel B at the top there's a 10-foot aquarium that was in my lounge room. We had 4 large individuals that we kept in that aquarium over a few years. And basically we would knock them out with an anaesthetic once a year and give them an injection with an antibiotic to mark their ear bones. And we would weigh and measure them each year and then return them to the aquarium and feed them. And there's a picture there of my son at the time when he was young, just turning on the local tap water. And so these animals were kept in pure fresh water at 500 metres elevation on the Atherton tablelands, and they were not kept in salt water at all. So the idea behind this piece of work was to demonstrate once and for all that this animal can grow and persist in freshwater without a need for salt water. At the time, a lot of the aquarium literature in some of the aquarium magazines from the Northern Hemisphere was suggesting that this animal really did need salt water. And this piece of work sort of definitively showed that that wasn't the case. And subsequently, you've seen this animal show up a fair bit in the aquarium trade in the last decade. Panel F just shows that they have really small teeth for a moray, and that led us to conclude that we thought we'd be able to keep this animal with other individuals of the same species in an aquarium without them killing each other because they had such small teeth. And that's pretty much how it turned out. It turned out that they liked eating fish, they quite liked eating earthworms, and they really liked eating live and defrosted prawn. They did fight with each other occasionally, and you can see in the little panel J down the bottom there, one of the individuals is trying to latch onto another individual. It's probably for a week or two each year they seem to razz up and fight a bit more than usual, and then the majority of the year they don't really have too much conflict in the aquarium, which is kind of neat if they're all roughly of the same body size. The interesting thing is when they did strike each other's body and scratch it, it leaves, you can't really make it out in panel K down the bottom there, but it leaves like a scouring pattern on their outer smooth surface, a bit like steel wool kind of scratching if you're cleaning a frying pan, if you're out camping or whatever. It leaves that sort of scratchy surface to it. And the really interesting thing is that they just slough that away and heal within about 36 hours. So even if they do have a bit of a contest, they don't seem to do any damage to each other. So the beauty of this piece of work, keeping them in the aquarium for a couple of years, was to, it showed not only do they only need fresh water, really, it also kind of led us to understand what they like eating and that they can cohabitat, which means they're a really cool aquarium animal because you can keep more than one of them if you've got a large enough tank. So in terms of what conditions they like, look, I think if you're going to grow them through to adulthood, you definitely don't want anything smaller than a large 6-foot aquarium. I think you probably want something bigger in the long run, especially if you're going to keep multiple individuals. Their temperature preference is really 24 to 26 degrees Celsius. That's their sweet spot when they're feeding really well and they don't show any signs of disease and what have you. So I think it's about 76 degrees Fahrenheit is probably the sort of temperature that you'd be looking to keep them at. Okay, a little bit about their feeding ecology. You don't need to look at the details here, but basically over a few years, I tried feeding this moray lots of different types of food. And as I said to you before, they really like earthworms. They really like prawns and shrimps and crabs and, various aquatic crustaceans. They don't seem to like insect larvae like dragonfly larvae and nymphs and that sort of thing. They don't seem to eat that, which is kind of surprising. They don't mind eating fish and they will hunt fish, but I've found that that's generally not their preference. They seem to really like the crustaceans and the earthworms, as I said. Interestingly, they don't seem to eat any of the freshwater molluscs that they cohabit with in these streams. They don't seem to like the nerite snails or the bivalves or even marine scallops or anything like that. And they don't really like eating some of the terrestrial meats like beef and ham and chicken and bacon. I did, after this work was published, I did eventually get one to eat a piece of ham on one occasion, but I can tell you that that's not something that they have to be pretty desperate to want that. So they are somewhat refined in their feeding habits. We've done a bunch of dissections on these animals to look at their anatomy when some of the individuals eventually died, and they have a really narrow, if you look in that panel just to the middle right, they have a blind stomach that's really quite narrow and small. Their mouth is their gape is fairly small, and the amount of food that they can consume today, in a day, is not a hell of a lot, to be honest with you, compared to some of the marine morays that I've kept. On the bottom graph there, we've got a few freshwater eels, Anguilla australis, Anguilla anguilla. There's published records on how much food they can eat in a day, which is around about sort of 4% body weight. And then with some of the marine morays, G. vicinus and Moringa, you see that they can kind of eat 12 to 15% of their body weight, those sort of amounts. So they're quite serious piscivores or predators. Whereas Gymnothorax polyurinodon, this freshwater moray, is in the middle of that graph. Most of the time, the maximum daily consumptions I'd observe for this species is around about 4 or 5% of their overall body weight. On one occasion, when the temperature was just right, we got some individuals to feed at about 9% of their body weight, but that's fairly atypical, I would imagine. Okay, so in the fish literature, you know, a lot of the time, because fish can't really handle and break their prey up, there's a lot of published work on their mouth sizes as an indicator of what they eat. And then more recently, there's been some pretty neat papers on how some animals use tools to crush large or hard prey open to overcome these limitations. This is just a graph from the Australian Wet Tropics from Brad Pusey and Mark Kennard and Angela Arthington's work, which sort of shows the mouth sizes of a whole bunch of freshwater fish in relation to their body lengths, with the body length being down on the horizontal x-axis and the mouth size being on the y-axis going up the page. And you can see that when I've overlaid it with the mouth sizes and the body lengths of G. polyaeronodon, you can see it's not a particularly serious predator. Its mouth size doesn't get that large compared to some other species. But because it gets so long eventually as it grows, it does become a fairly decent predator, I guess. But I'd argue that by and large for most of their life, they're a meso predator. They're eating sort of small, medium-sized prawns and shrimps, that sort of thing. But their feeding ecology, what is interesting about it is how they've got some party tricks that take them to the next level. Okay, and that's partly because they have a second set of jaws sitting down in their neck. And that's the really cool thing about morays. So courtesy of the Cairns Base Hospital, what you're seeing here is a CT scan of the upper body of a moray. So its head's at the very top and its jaws, and you can see just come down a bit, that second set of jaws that's down below the head, which floats in the neck next to the spine. And that second set of jaws that this animal has tucked down in its neck is actually, you know, quite a serious jaw structure. And so they use that to actually grab their prey with the front mouth, with their mouth, and then these second set of jaws come through and grab that food item, and then the front mouth opens up and releases, and they use that set of jaws to drag the food down their throat. Now, keep in mind that this animal is feeding by poking down into interstitial spaces in the rocks, and so it can't necessarily open its mouth in a really wide fashion in the way that some of the more serious predatory eels and fish could do. And so then when it's in a confined space, it's able to use this second set of jaws to transport the food down through its neck. So quite a cool party trick. Yeah, and now I'd just like to show you one more video as a closing, and I think you might enjoy this one. So if you just bear with me for a moment, I'll go on. Give this one a run. This is pretty neat. So, you know, you get to keep this animal in captivity, and you get to see some things that, well, let's just say they're a bit out there. So just take one last look at those pharyngeal jaws, that second set of jaws that's sitting down the neck there of the eel in that CT scan, and then try and imagine that working in this next video as we have a look at how the freshwater moray actually feeds in captivity. So this first individual you're looking at, her name's Monique. At this point in time, she's probably about 80 centimetres long. She's a fairly big adult. She's just eating an earthworm there. She's pretty hungry, having not fed for a couple of weeks. And here she is. She's just going to show you one of her little initial warm-up tricks. So finds that earthworm, grabs it, does a little bit of a spin. She's pretty excitable on this occasion. She doesn't really need to do that. She can just consume that earthworm pretty easily because it's a small prey item. Now here these two individuals are feeding on larger prey items. These are defrosted marine prawns. And just watch that neck working, that second set of jaws. You can see that tension as it works between front and second jaw. And you'll see on this second individual the same process again. You see that real puffing back of the neck and the second jaw working hard to move the food down the throat. Now, this is a really large prey item for this individual to take on. This eel, her name was Black Sally. She's a really nice dark coloured individual. and she's probably about 80 or 90 centimetres long at this point. She's going to spin really rapidly here. So she rotates about 10 times in the clockwise direction and then about 10 times in the anti-clockwise direction, all within the matter of a couple of seconds. And what she's doing there is trying to smash that prey item on the bottom and break it up into smaller bits. Now, this one here is called Wilbert Pelican, and he's doing an amazing little party trick in terms of breaking his food up. You can see that big piece of prawn that he's working on. He just breaks a small piece of it off there in the knot. And then Cedric comes along and eats the lion's share of the food item. Now, that'll happen pretty quickly. So I'll just show it to you in slow motion replay. We have a look at what Wilbert Pelican's doing here. He's just found the food. He's grabbed onto it, got a good grip, and starting to go into that spinning, rolling motion, but chooses not to use that strategy and then opts for the knotting behaviour. You see this knotting comes from the back of the animal, from the tail, and it's a very loose knot on that first occasion. Now, this time the knot's going to get a lot tighter. You can see there's a real constriction going on. It's getting tighter and tighter. And watch just here now. There'll be a real tight squeeze of this food item as it pulls it down there and tears a very small piece off and eats it. And then Cedric comes in over the top and takes the majority of the food item. So it just goes to show you that these freshwater moray eels, they don't have simple feeding mechanisms. They have multiple strategies available to them, with this second set of jaws to move food down their throat, with the spinning and smashing action that they can use on the surface, and then this higher level squeezing, knotting action. Because they don't have hands like we do to use a knife and fork, they've really just got the mouth and their body. And so it's just a fascinating little combination of strategies that they've got available to them for feeding purposes. I hope you enjoyed having a look at that footage. Brings back fond memories for me. And so just to finish off here on the freshwater moray, there's a few papers that you can get access to read more about the biology of this animal if you're interested in the science side of it. One of those papers there, oh no, it's actually not even up there. There's another more recent paper which goes into some of the aquarium care of this species. It's worth tracking down as well from some of the work I've just showed you. Okay. So thanks to just 1 million people and organisations that have helped out with access and various things over the time. I'm sure I've left a few out from there, but my apologies if I have. Thanks to everyone involved and the few times we've got a few grants to do some of this work. And, you know, I really appreciate TropWATER's ongoing interest in this type of work in the Australian Wet Tropics. So thanks again, and I guess I'll throw it over to you guys for some questions.
There we go. I think you can hear us. If I can get this going the right way. There we go. That's what I wanted to do. So can you hear me okay, Eb?
Yeah, loud and clear, Michael. Thanks, mate.
Perfect. Yeah, thank you. The presentation was definitely interesting, if I don't have to kick my desk. And I'm just going to give it like 10 seconds because, like I said, the YouTube has that slight delay, and let everyone get their questions in. And it looks like we actually already have our first one from Rich Byerly, and he was asking if the eels have been exported.
Well, definitely this species of eel is exported in the trade. I think the animals are by and large not sourced from Australia. We don't have large populations of this species. We've definitely got freshwater morays coming into the country that are being collected elsewhere in the Central Pacific. So this species is probably a bit more abundant in some other parts of sort of Indonesia, Papua New Guinea. I've definitely seen them in the Solomon Islands and you know, in some parts of even continental Asia. So yeah, they're in the trade. They go for a pretty decent price these days. In Australia, where obviously they've got to be collected under permit, so they're not something that is probably being traded too much at the moment. But you know, you'd definitely be able to buy some over the internet. Yeah.
Awesome. And I know during the video in the chat, they were talking a lot about, especially with the eel, whether or not they've been bred in captivity, and there was some kind of discussion. Can you touch on that a little bit about breeding them in captivity?
Well, I haven't been able to breed them in captivity. I've certainly had the females rowing up every year in what's our wet season, so sort of in the hottest part of the year here. So the females definitely, they get really, really gravid. You know, they almost don't look like an eel. They end up looking like a cod. You know, they get a really fat belly. When we've had a few individuals die eventually and done autopsies on them, they have hundreds of thousands of eggs. The eggs are not super small, so I'm not aware of anyone breeding them in captivity at the moment. But I think it'd be quite possible because the eggs aren't especially small. And you know, given there's aquarists out there now and commercial companies breeding a lot of the marine species with really small eggs and larval phases, I think it's quite possible. Just keeping in mind that, you know, the larvae are going to spend a considerable time in seawater. You know, they're not going to be a freshwater, complete their life cycle species. So, yeah, and given they're a fairly high value animal and really attractive and really interesting to keep, I'd be very surprised if a company doesn't move on to trying to commercially breed them. I don't know,
definitely,
Michael. Sorry.
No, you're good. Like I said, I like the comments there. And then Becca Teen is saying, hi, Eb. That's Eric Thomas. That's his YouTube name, if you didn't notice that. And he was asking, do any researchers work on goby reproduction / husbandry / rearing?
Yeah, it's a really good question, mate. I'm not aware of any commercial groups working on it, but I don't really move in those circles. In terms of your actual question about researchers, there's a couple of papers just coming out now, and there's a couple more in review from particularly some of the French scientists starting to do experiments on what water quality parameters they require, because their life cycle is quite tricky from hatch to going to the ocean as like a 1-2 day old larva, and then coming back a few weeks to a couple of months later, and they're still quite small at that stage when they're coming back into the rivers. And they sort of, the bit of work that's been done so far has sort of suggested within 24, 48 hours, they undergo a really radical transformation to basically have the same characteristics as a juvenile as they move from the estuary then back into the fresh water. So, yeah, there's some really cool science from Midori Aida from Japan's done some stuff on the temperate Stichiopterus goby, and then a couple of the French scientists, Niels Tykart and co, are doing some really neat stuff on their water quality. And it's kind of showing they need really specific salinity levels at the point that they're transforming. So I think the science is already being laid down. So I think as people come in to try and rear them in salt water as larvae and then transition them back to fresh, I think a lot of the knowledge is kind of out there or is coming out there now from the researchers. So I don't think it's going to be long until you see the full cycle being completed for a whole bunch of these pretty little cling gobies, rather than having the wild collection based strategy which has been predominant in the aquarium trade up until now.
Awesome. And I think that question's kind of the same answered there by Josh Black, asking if the cling gobies have been bred in captivity.
Yeah, I haven't heard people like hobbyists doing it at the moment. I know that I think we might be up to around about 180, 200 species of cling goby in this subfamily, which is, I hope it's not too over the top. Science name is Cicidea gobies. So in that group, there's probably 180, 200 species now that are described. I'm aware of 2 of those species do not have to have a marine larval life cycle, so they can complete their entire life history within fresh water. So I would have thought that they'd be good candidates to start on. But as for all these other species, yeah, I'm not aware of people pulling it off in captivity as yet. But, you know, given what people are now doing, marine hobbyists, both amateur and professional are now doing with a lot of the reef fishes, I think it's just a matter of time. So just reiterating, keep in mind that almost all of these cling gobies have a marine larval phase, and then they have a juvenile and an adult freshwater phase. So that obviously adds a little bit of a degree of complexity to completing the breeding cycle in captivity.
Yeah, it definitely adds a challenge in there. And then Rich Byerly was asking if you've worked with either Matt McGee or Peter Unmack. He was just curious.
Yeah, I work a bit with Pete Unmack. He's obviously spent a lot of time over in the States, and he learned all your magic tricks and brought them back over here to us. Pete's doing amazing stuff around Australia at the moment with a lot of our rare and threatened freshwater species. And so whenever they're in my neck of the woods, Pete gives me a ring, and I come out and drag a seine net with him, and we look at a couple of the rarer rainbow fish in the region, which we've got a few that are, quite exquisite and are in a fair bit of trouble. Some of them are really in trouble in terms of populations being reduced and their distributions sort of eroding before our very eyes at the moment. But that's quite different to the freshwater cling gobies, because obviously, as you guys all know, breeding rainbow fish in captivity is really easy. And, so it's kind of neat in their case that if we can spread them through the hobby, then we've got that extra arc population sitting in America or Australia that gives us some reassurance that those animals are not going to fall off the back of the planet, So, yeah, Pete does some great work. I haven't worked with Matt. So, yeah, thanks for the question, Rich.
Yeah, and then I'm just throwing this out there. You were talking about Rich's fish room tour before the meeting. You mentioned you were watching his tour. This is the same Rich Byerly that you were looking at. So I know you were very impressed with his fish room. I figured Rich would get a kick out of that, and just wanted to mention it.
Yeah, actually, Mike, if you don't mind me saying, Rich, I really did enjoy going through all the tanks, and I'd sort of done this. I don't have nearly as many aquaria as you. I'm not sure anyone in the world does. But I was out in my aquarium shed filming it and just showing you a few of the animals I've got to splice into this. But because I'm technically so retarded, I'm down in Townsville now, and I had to reshoot the video in my cousin's bedroom here with the Kookaburra picture behind me, and so I didn't get to show any fish room stuff. But it's great. You got a thriving society with people keeping lots of animals, and obviously that's how most of us learn about stuff and what they can do. It's really cool.
Definitely. Rich has been a good member of our club. Very interesting to have. Lots of knowledge. And C.A. Benner was asking for the Goby habitat pre-data pre and post 2015 that showed a decrease in the percentage of favorable habitat post 2015. Was this due to environmental change?
Yeah, look, really good question. I probably was a bit unclear in a lot of stuff that I said. I was watching the video back just then. I only filmed that like a few hours ago, and I definitely missed a few things. I didn't explain that very well. It wasn't because of loss. It's not because of a loss of habitat. It's not because suddenly the wet tropics is deteriorating because of, urban sprawl or agricultural development or something like that. It's simply because the data that we put into the model initially was from a lot of our ad hoc work, where we just kind of went and snorkeled as many places as we could on weekends and willy-nilly and patched it all together and put it into the model. And I thought what would happen when we got paid by the government for a year to go and snorkel those 12 or 13 streams and then add that to the model as well to update it, I thought what it would say is, okay, now we're finding cling gobies in more streams because we're looking in more streams, which means the model will tell us that, they're actually not as rare as we thought. They're going to be in more and more places. But actually what it did was to say that a number of those regions we sampled, they're not in those regions. And a number of the drier sections of the wet tropics, they're not thriving in those locations. And so actually where we'd found them initially is really they're only in a few of those really wetter places. So the model came back and told us, yeah, you've put in a whole lot more work, but that's actually telling us that our projections say that they're actually in fewer places across the region than we would have expected. And that's just because we had to sample it in a more systematic way, which told us basically that our intuition was right initially. I'm not trying to sound like I know everything. It was like the places we found them initially was pretty much the places where they are. And the further afar we looked, we didn't find a lot more population. So I guess it probably is really just showing my lack of understanding of how predictive models work, more than anything. So, yeah, if you wanted a succinct answer, you've got the wrong person.
Yeah, I know how that goes for sure. Let's see. Finding the next question. Josh Black was asking, what's the best diet to feed cling gobies?
Okay, Josh, good question. I didn't have time to go through all those different groups of cling gobies. So even though a few of them look pretty similar, there's probably about half the species I showed you are just true herbivores that graze on the algae on the bottom. They don't eat like macrophytes. They don't eat the aquatic plants you'd have growing in your aquarium. They just eat those algal slimes on the rocks, the microscopic stuff with the diatoms in it and whatever. All of the ones in the group called the Stiphodon genus, they're all just little grazers. So they just basically look like a tadpole, and their little teeth are just like little toothbrush combs, and they're just going along like that all day. So I found keeping them in an aquaria, the key is really really good lighting and good biofilms. And I think they tend to do better in tanks without a mixed community type thing initially, until you get a feel for them. They do really well in established tanks, and you've got to be a bit careful when you've got planted out tanks, because that's obviously competing with that algal biofilm. I had most success with the bigger herbivorous cling gobies in sort of big aquaria with good water quality, with partial sunlight for the day, which is obviously a killer for most people, keeping their tanks clean and keeping the front glass looking nice. But if they get sort of 4 or 5 hours direct sunlight, I think they did really well, most of them. Some of the Stiphodon group will also eat bloodworm and, frozen zooplankton that's defrosted, and, the usual, they'll like a bit of that stuff. The rabbit-headed cling goby, the one I think we should be keeping more of in Australia as Aquarius, it is a strict biofilm feeder. It doesn't like even bloodworm, which is quite surprising. Some of the other group, the Smilosicyopus and the Cyciopus groups that are above the waterfalls, they eat insect larvae, aquatic insect larvae, and little tiny baby shrimps. You know how you got your little cherry shrimp sort of size stuff that you keep in the trade. They're eating the little larvae and juvenile of those shrimps. So, they're not eating the adults, which are, 1 centimeter, 2 centimeters long. They're eating the 6, 7 centimeter long young of those shrimps. So you can get them onto bloodworm and, even little bits of ham, and, you can cross them across to the usual frozen diets pretty easily. Yeah. So, yeah, it really depends on the type of cling goby you got.
Definitely. And then, let's see, Larry Schenkel was asking if you've ever worked with Gary Lang.
I haven't worked with Gary, but he's come out here a number of times as a guest speaker about rainbow fish stuff to our, I guess, which would be like the equivalent of your Sacramento Fish Aquarium Society. We have, you might have heard of ANCFA, which is the Australian and New Guinea Fish Association. And sort of he's come out, and I've met him at the ANCFA functions, and he's obviously a wealth of knowledge. And, you only have to be on YouTube for 3 minutes to work out that he's kind of like the president of the rainbow fish world. So it's kind of cool that there's an American championing a group of Australian and Papua New Guinea fish. I think I like that we finally had a group of fish that's broken into the aquarium trade at the global level. So us little underdog Aussies, I think it's a good news story. And he's obviously a sweet person with a lovely, warm attitude that is about sharing and spreading knowledge and being inclusive. So I think that's sort of guy like Gary Lang. It's just wonderful to see pasted all over the internet,
Definitely. And then Josh Black was asking, when did Australia list the cling gobies? Was it recently? He was curious how Stiphodon, I'm pronouncing that wrong, I know it, got into the hobby. He's only started seeing them in the last decade over here.
Yeah, look, I'd say pretty much everything you've seen in the hobby will not be coming from Australia, because our populations, as I said in my presentation, are just so small. We're really only talking of tens or hundreds of animals. And so I was involved in getting a few of the species listed in Australia initially, because my concerns were that as an aquarist myself, I really want to keep these animals. They are beautiful. They're really cool in aquaria. And I copped a bit of a backlash from some of the hobbyists in our country here for getting, being involved in having them listed. But I think anyone there are a few streams out there in Australia that actually have a good few hundred of some species in them, and then a year or two later they won't be there, and then they're back again as their larvae arrive, So I don't think Australia is a great place to be harvesting this species for the trade, especially when we haven't completed their life cycles. But there are places in the Pacific, on the Pacific Islands, where some of these species are really abundant. So I've been in some streams where you've got tens of adults per square meter, okay, in the stream. So they're really arriving like locusts, and they're in plagues. And I'd say a fair bit of stuff that you're getting in Europe and America in the aquarium trade is being harvested from those places where they're in high numbers and they're easy to harvest, and you potentially not maybe over harvesting them because they're real production places. I'm real pro the idea of keeping things in the aquarium trade, and I love keeping things. And I just think, just in the Australian context, I think we should be able to have a sustainable quota for the rabbit-headed cling-goby, as I said, which is a bit more abundant and widespread. And it's actually can be quite hard to catch at times because they're quite fast and wily. But some of the Stiphodon group are really easy to catch. I know that people are legally harvesting them, sneaking in and catching them at times where there's really small populations, and it's really hard to enforce that, So yeah, I think, wild harvest from places where it's a sustainable practice and they're really abundant. And I'd love to see us complete that circle and maybe get a bit of private enterprise and government sort of investment in trying to complete the cycle so we can all enjoy keeping those beautiful animals in the hobby,
Definitely. And then real quick, before I go into the next comment, I want to just say last call for questions if anyone's got them. I know you're on a limited time frame. We've got about 10 minutes left of your time, and just wanted to throw that out there before I start the next thing, since there's that delay on the YouTube end. And then I also want to shout out our Teespring promo code. It's 20% off through this weekend only. The code is up on the screen. It's been dropped in the chat a bunch of times. So we've got lots of great things up on our Teespring store, which the website is there now on the screen. And let's see. I am looking to see if there's... What was that?
Sorry to butt in, mate. I didn't actually answer that question. I think the Stiphodon semini has been listed in Australia for probably, I'm guessing it's probably been listed as critically endangered for 7 or 8 years now. There are 3 other Stiphodon species that are listed are not listed nationally. They're only listed under state legislation in Queensland. But all of the Cystidean gobies in Australia, or in Queensland, because they're only, at the moment, we've only found them in Queensland, none of the other states of Australia. They're all listed as no-take species, which doesn't give them necessarily all threatened species status, but it means that you're not allowed to just go and harvest them and pop them in your fish tank. So, it's quite complicated. They each have different levels of status, but most of the action's been in the last 5 to 8 years in that regard. And I think some of the listings should be changed now as we know a little bit more about them. But it's up to people from the public or some of us as scientists to put in the time to update those listings.
Definitely. And yeah, no worries for butting in. That's not a problem. It looks like we don't have any more questions. Everyone's just saying thank you for coming out. It's been a super fun talk and super informative. And we really appreciate it. We know it's kind of, you're squeezing us into your schedule, and we really do appreciate it. So is there anything else you want to say before we log off here?
No, Mike, you've been great. Everyone behind the scenes has been really helpful. And I'm not very busy or important. It's just that I got a 6-hour drive back to home tonight with my wife, and it's Sunday night for us over here, and that's the only reason I'm leaving, not because I'm important. And the bits and pieces I've heard from your society and the talks that were taped, just fantastic. Love listening to it. So big shout out to you guys at the Sacramento Aquarium Society. You do a great job.
Awesome. We appreciate that. And so we're going to go ahead and bring Josh back into the stream here. And if I can put him here. And then me and Eb are going to kind of go back behind the scenes and let Josh go ahead and close it out.
All right. Hello, everybody. Once again, we want to thank Eb for that great presentation. It was really interesting and very informative. I didn't know so much about the cling gobies, and now I definitely feel like I've learned a lot more. So yeah, thank you, Eb. Best of luck with everything. And again, our Teespring code is good until midnight of tomorrow. So feel free to purchase anything for 20% off. Your promo code is right there. It's R2M7A5CX8Q. So yeah, if you're interested, do that. Feel free to, once again, donate to our club if you want to. You can do that at our website down there. And also feel free to join us in membership. And that's also, the link is also on our webpage. I don't have a whole lot of other announcements at the moment. Just once again, thanks for joining us. And we hope to, oh yeah, congratulations to all the new Board of Governors and all of the committee people. And looking forward to meeting all of you and continuing our great work we've been doing. And we're looking forward to next month's talk, and that would be, let's see. Well, it's on the website. I apologize. So go to our website. We'll have more information. Check out our Facebook page. It's all on there. But we look forward to next month's meeting, and I hope you all have a great rest of your November and Thanksgiving. Happy Thanksgiving.